Molecular techniques. Molecular diagnostics represents a significant advancement in clinical microbiology, and it's considered by many as the fastest growing section in many clinical laboratories. It provides clinicians with rapid answers for treatment options and saves valuable time in cases of life-threatening infection. So, blood infections, sepsis. If we have a blood culture tube on a patient and it comes down to the lab and we put it on our system and our system detects that it's positive for growth of microorganisms, what we used to have to do is we would gram stain it and report the results directly to the physician, and then we would have to culture it. And culturing it takes anywhere from 1 up to 3 days to get results on, especially a little bit longer if it's going to be a anaerobic pathogen. So we know the gram stain result, and the doctor can kind of start treatment a little bit, but we don't really know the causative agent, which microorganism is causing the infection, for about 1 to 3 days. With the advancement in molecular diagnostics, we can take a small drop of that infected blood, put it into a molecular diagnostic tool, and they have different types of systems that can identify the genetic material of different types of organisms, and we can get an identification within several hours. So instead of waiting a day to 3 days, we can get that information, that diagnostic information, finalized and sent up to the doctor sooner. Techniques for identifying organisms through molecular diagnostics. Nucleic acid hybridization. So this is the formation of hydrogen bonds between nucleotides of single strands of RNA or DNA that are complementary to each other. Under the right conditions, it results in double-stranded hybrids. So we can have DNA-DNA hybrids, DNA-RNA hybrids, or RNA-RNA hybrids, also called duplex formation. Terms associated with the 2 single-stranded nucleic acid molecules. So the target is the template, that's what we're looking for. That's the sequence that's going to be identified. It's immobilized or suspended in solution. The probe is specifically designed to identify the target. So if you remember your bases, A pairs with T, G pairs with C. If we have A as the target we want to look for, the probe would be T. So it would be the complementary strand to that target. It's a single-stranded RNA or DNA oligonucleotide labeled with a reporter molecule, chemical, radionucleotide, or fluorescent particle. So the probe is labeled with something that we can detect. So we wash the DNA sample with the probe, and if the probe finds its complementary strand, it's going to stick, it's going to bind, and then we can evaluate or try to detect that reporter molecule. If the reporter molecule is present, then we know that that specific sequence we were looking for is present, and that's how we can diagnose disease. So we can say we're looking for the genetic material of syphilis, and syphilis is the only organism, to our knowledge, that has this sequence. We can develop a particular probe against that sequence. We can wash the DNA from the organism we've isolated with that sequence, and if we see the reporter molecule when we do our evaluation, then we know that that sequence was found. We know that that's the organism causing the disease. So here is just kind of the steps. So we have our double-stranded DNA. This is from our organism that we're trying to evaluate. Then we have the gene of interest within that double-stranded DNA. So in order to get the probes to stick to the DNA, we need to separate those strands. So we're going to denature it. Denaturing just means to separate the strands. So now we've got 2 single strands, and we're going to add our DNA probes. The DNA probes are labeled with some type of fluorescent molecule, molecular beacon, radioactive molecule, something that we can detect by our instrumentation. Then we're going to put the probes in the presence of that DNA, and the probe has a very specific complementary sequence to the gene that we're looking for. So once we wash it with the DNA, the probe is going to find that sequence if it's present, if this is the correct organism in the patient sample that we're looking for. It's going to find that specific sequence and bind, and then we're going to be able to detect that binding through our instrumentation. Hybridization reactions has a lot of variables. It's very sensitive and specific, so it's kind of picky to get the correct results. First of all, the temperature. In order to denature that DNA or separate the strands, we got to make sure that we have the appropriate melting temperature. If we know that our DNA contains a lot of C's and G's, we're going to have a much longer or a much higher melting temperature because it takes a lot more to separate strands of DNA that have a lot of C's and G's. The length of the probe. If we have really long probes, it's going to be tough to get that really long probe into the DNA. Short probes maybe not as effective either because there's the more bases you can have in a probe, the more specific that probe becomes for your target sequence. It's pretty likely that every organism on Earth has the sequence ATTCG somewhere in it. It's more unlikely that every organism is going to have ATTCG, ATC, so forth. So however long we can make that, it makes it more specific for that particular organism that we're looking for. Probe concentration is another one. We got to make sure that there's enough probe. Too much probe or not enough probe, both of those promote nonspecific binding. Or excessive probe concentrations promote nonspecific binding. Not enough will not allow for binding at all. The degree of target and probe complementarity, so we want to make sure that it's exact. If mutations happen in the host organism, then the probe isn't going to exactly match that target sequence. Salt concentrations impact the ability to bind. pH also impacts the ability to bind. Probe selection is very important. It determines the recognition of the target sequence. It forms a duplex with every complementary sequence available for the reaction. It can be DNA or RNA based, and they're labeled. They're labeled with something that we can detect, either some type of fluorescent molecule or something that we can pick up by our instrumentation. So that's one method. We extract the genetic material and we use a probe to specifically find where, if that probe will bind to its target sequence. We want the target sequence to be specific for a microorganism, and if we have the probe binding to our patient specimen, we know that it's binding to that specific sequence we were looking for. We know the patient's positive. The other thing we can do is nucleic acid amplification procedures. So amplification may be used to increase the target microorganism's nucleic acid in the sample in a short amount of time. 3 assays typically have high sensitivity and specificity. It requires specialized testing and detection equipment, dedicated space, and specifically trained scientists. So polymerase chain reaction, PCR, was originally described in 1971. It can amplify a single copy of a target DNA into exponential amounts of nucleic acid product over 25 to 40 reaction cycles. PCR is simple, sensitive, and powerful. So what happens during PCR? So we have 2 primers. One primer is a forward primer. The other primer is a reverse primer. What happens with these forward and reverse primers is, first, we separate the DNA, denaturation. So now we've got 2 separate strands of the DNA. Then we have annealing. So that's when we want to bind the primers to the DNA. So when the primer binds, it's going to stick to these regions of the DNA. And it's a very specific region of DNA, kind of like when we talked about the probes previously. Primers are going to bind to and recognize a specific region of DNA. And we design these primers to be adjacent to genes of interest. So if we know we want to amplify a particular gene, we're going to put the forward primer at the beginning of the gene. We're going to put the reverse primer at the end of the gene. And then those primers are going to copy by moving forward or backwards, depending on if it's the forward primer or reverse primer. So basically they're going to go in opposite directions of each other. One's forward, one's backwards. And then it's going to fall off, and we can make many copies of that centralized region based on this process. So once the primers bind, this primer, the reverse primer, is going to synthesize DNA in that direction. The forward primer is going to synthesize DNA in that direction. And then we're going to continue this process where that centralized region is going to keep getting amplified and amplified and amplified until we have a lot of that centralized region. And that's our gene of interest that's in the middle. So denaturation, primer annealing, and primer extension, those are the three basic steps. So here is another diagram of that process. We've got our double-stranded DNA. Denaturation, we separate them. Annealing, we have the primers bind. We have a forward primer and a reverse primer. Then we have the elongation, which is the third step. So the primers are going to then elicit copying of the rest of that gene of interest, and we're going to create these centralized segments. And those centralized segments are going to continue to be amplified and amplified and amplified until we have tons and tons and tons of them. So the template DNA is generally from a sample. The primers are generally 15 to 30 nucleotides in length. Thermostable DNA polymerase, Taq polymerase is the one we usually use. We need a polymerase to help make the DNA during the elongation process. Magnesium chloride is required for the function of the Taq polymerase. There's a buffer to prevent changes in pH. Deoxynucleotides, so we need D and TPs. All four bases need to be present. If we're going to be making DNA, we need to have bases there. And then thermocycler, it's not necessary, but it sure makes things a lot easier. They used to use different temperature blocks where they would do the annealing at this temperature block, and then they would have to move it to the next temperature block for the, denaturation at one, annealing at the other, and then elongation at the other. But now we have a thermocycler, which is a block that changes temperatures automatically according to the cycles, and it can cycle this many times for us. So you can pause the video and read through what each one of these is and what it does in this reaction. You will be tested on these individual components of PCR. This is pretty essential and vital to the understanding of how molecular diagnostics plays a role in identifying bacteria. So that's something that you want to do. Contamination prevention, so PCR is extremely sensitive. It'll amplify anything that has that sequence. So if you have contaminating DNA, then it might amplify something from the environment instead of amplifying something that was actually in the patient specimen. So there's specific work areas or workstations that you work in. Some labs require you to shower before you even come near the PCR station. There's special lab coats you have to wear. Hair has to be tied back. So there's all different precautions that are taken when preparing specimens for PCR. PCR product analysis. So usually what would happen is we'd do the PCR, which takes about 4 hours, and then we would have to run the specimen out on a gel, an agarose gel. And this is what that looks like. So there's a positive side on one side, a negative on the other, and basically we would put all of the DNA on the negative side because DNA has a negative charge, and then we would run to red, which means that when you turn it on, there's a negative charge floating in this chamber, and since the DNA has a negative charge, negative and negative repel, so the DNA is going to move through the gel because it wants to get away from that negative charge. Then we have a positive charge over here, and negative and positive attract each other. So the DNA is going to want to migrate through the gel toward the positively charged area of the gel, and that will separate DNA by size. So smaller DNA can go through a lot faster. Bigger DNA takes a little bit longer to get through all of that porous gel material. So that's how we separate DNA by size using charge in gel electrophoresis. The problem is it takes 1 to 2 hours to separate these PCR products. It could generate hazardous waste if you use certain types of stains, like ethidium bromide is a carcinogen. So if you use that in order to visualize DNA at the end, that's hazardous. And it requires an imaging system. So once you run it on a gel, this is literally what it looks like. everything looks blue. That doesn't mean that there's DNA there. So you actually have to look at it you have to stain it with something and then look at it using UV typically. And you can use the UV and capture a picture of the gel. So this is a picture of ethidium bromide stained PCR amplicons. So there's different patient specimens. This first well here is just a marker, and this marker is of known base pair lengths. So like this marker right here says that this is exactly 400 base pairs. This marker here is a 100 base pair marker. So the dye front is here, and then if the patient had the HPV, that you would see that amplicon here. So here is, usually they do like a negative control, a positive control, and then if the patient specimen has it, it will have a band in the same area of the gel as the positive control. So that you can see how you would evaluate this. Now we have PCR primers that can not only identify different species, but also identify like resistance markers. Like we have the MEK-A gene, which gives staph species methicillin resistance. So if you've ever heard of MRSA, methicillin resistant, Staphylococcus aureus, we have primers that will recognize the gene for that methicillin resistance, the MEK-A gene. And if we do this PCR process and run it out on a gel and we see a band where the positive control is in our patient specimen, then we know that the patient has that MRSA. So again, we usually have a negative control marker, a negative control, a positive control, and then we have multiple patient specimens that we can run on here. It turns out that this was the only patient that was actually infected or actually positive. Here's another one that you can see what they look like with different patient specimens. But instead of running it out on a gel, which isn't commonly performed today, we do have real-time PCR. PCR amplicons are detected as they are accumulated after each cycle. The reactions occur in closed tubes that do not have to be opened, which reduces the likelihood of contamination. Analyze fluorescence, so fluorescence per cycle, and then we can analyze the melting curve to detect or to determine results or verify purity of PCR product and determine whether the primer dimers are present. So in real-time PCR data analysis for herpes simplex virus, this is just a chart that shows cycle numbers, and you can see the fluorescence increasing with the cycle numbers. So the longer we let the PCR run, the more we see it increasing with the fluorescence. This is a picture of one of the older systems that we used to use for real-time PCR. We have more modern systems today. And there's many ways to detect real-time PCR, but one of the easiest to explain, since this is microbiology and not our molecular diagnostics class, is a molecular beacon. So what a molecular beacon is, it has this fluorescence molecule and a quencher. Anytime the quencher is next to the fluorescent molecule will not be able to glow or produce fluorescence. If we amplify a region of DNA, we have the complementary PCR product up here. So what will happen is this thing will unfold and stick to the DNA. And when it does that, the complementary region binds to the PCR amplicon. So this is what we just amplified by PCR. The quencher moves away from the fluorescent molecule, and this is allowed to fluoresce. So this will fluoresce and produce a light, and then we can detect that fluorescence. So initially, going back to this, you're not going to see any fluorescence. Even though we are amplifying the gene of interest, it's not enough to fluoresce enough to be detected by this system. But once we get to 20, 25 cycles, now we're going to see a lot more fluorescence. And if we see that fluorescence, we can safely assume that our molecular beacon has bound to the gene of interest and that our patient sample has that gene that we're looking for in it. There's also cyber green fluorescent dye that binds to double-stranded DNA. The problem with this is that just because it's binding to double-stranded DNA doesn't mean that it's the DNA of interest. It doesn't mean that, so it's pretty non-specific. It's going to bind to any double-stranded DNA product. So again, advantages, it may be used for the melting curve analysis. It's cheaper than using the labeled products, but again, disadvantage, it binds to any double-stranded DNA. Many companies produce kit-based assays, so now when you go work in microbiology, if you are doing a molecular test to diagnose a particular pathogen, you put the patient's sample directly into a cartridge, and then you put it on the analyzer, and it will do everything for you. So it will pipette different reagents into the different compartments. You never have to open up the equipment. It's all done internally. It reduces the likelihood of contamination and reduces the likelihood of error because it's all automated. So as the tech, all you do is inject the specimen where it needs to go, put it on the system and press start. And then the system will actually go through all those steps. If those systems ever break down, it's your responsibility to know what all of those steps, what was happening during all those steps, and how to kind of pick up the pieces and continue on with the diagnostics if the automated system is broke. So that's why we go through the basics of molecular diagnostics in next semester's class. So this semester we focus on just basic overview in clinical microbiology, but once we get to actual molecular diagnostics, we're going to learn more of the nitty-gritty details so that if an analyzer does break, you'll be able to pick up the pieces and perform the tests. Reverse transcription PCR, RT-PCR, detects mRNA products. RNA transcripts from a single cell are possible to detect. You must extract the mRNA though. Reverse transcriptase, RNA-dependent DNA polymerase, generates complementary DNA, and then that can be used to detect RNA viruses. You can detect mRNA products, reverse transcriptase RNA, this is a repeat of previous. Multiplexed PCR. So multiplex PCR is a new and emerging, really cool idea. So this doesn't have clinical applications yet per se, but it will in the very near future. So what they can do is take a swab from a patient's nose, nasal swab, or a respiratory swab. They could swab their throat, and then they can do a multiplexed PCR on it to identify any possible microorganism that could be causing the infection. So we have a list. Let's say these are the top 10 most likely respiratory pathogens. Now we have primers for each one of those top 10 respiratory pathogens. So because we have multiple primers in there, we can identify 2 or more targets from the same tube. It employs different primer sets. So it has these primers for this pathogen, these for this pathogen, these for this pathogen. So if one of those 10 pathogens happens to be present in that specimen, the PCR will amplify it, and you will be able to see the results of that. It can be adapted to real-time PCR or analyzed by agarose gel electrophoresis. Reaction conditions must be similar for all targets, and the primers must have similar TMs. So we have to make sure that there's similar conditions, otherwise, because we can't incubate the specimen at different temperatures and times for different organisms. So they have to be kind of similar genetically so that we can perform this test. But they have found a lot higher sensitivity and specificity using these multiplexed PCRs compared to like standard culture methods. So instead of plating them on the 50 different plates and spending the money on all that, you spend the money on the molecular diagnostics, and it has equal to or greater than specificity and sensitivity than our traditional culture methods. So this is what a sample multiplex PCR would look like by gel electrophoresis. Nested PCR consists of 2 different consecutive PCR assays. So the standard PCR amplifies a target region, and then the nested PCR amplifies a portion of the target region. So the first step is to amplify a big region, saying this is the gene for this. Then the second step would be to amplify a more specific region within that gene. So now we know we have this mutation within this gene here. So We nest, it's not multiplexed, it's not identifying different DNA structures altogether. It's like kind of like those nesting dolls. It's one smaller PCR product from within a larger PCR product. So as an example, the first assay might amplify whether herpes is present or not. The second assay might amplify a region that distinguishes HSV1 from HSV2. The drawback is there is potential contamination when opening up the tube. Other nucleic acid amplification reactions, strain typing and identification. So the importance of strain typing and identification are important because of antimicrobial resistant organisms. It increases the rates of toxin-producing bacteria, or increases in the rates of toxin-producing bacteria if we know it's a specific species and we know that that species has been producing toxins or has mutated to be able to produce toxins, and that's now becoming a public health concern. Knowing the exact identification is essential. And the spread of pathogenic microbes worldwide. There is a need for accurate epidemiological surveillance of these organisms. Future trends in molecular diagnostics, so procedures are becoming cheaper, easier to use, and more available. Increased use in clinical microlabs is expected. Advances in the sequencing of microorganisms provide much potential for future laboratory methods. Currently trending standardization of molecular diagnostic methods and kit-based detection products. So sequencing is getting the order of nucleotides in DNA. There's many different methods for sequencing. The Sanger method is chain termination. 16S rRNA gene sequencing identifies isolates and determines relatedness. Chain termination method uses nucleotides that terminate the string with a fluorescent color. Fragments can be aligned by size and last fluorescent nucleotide. There's dentograms, a rough prediction of similarity based on the comparison of sequence structure, the relatedness or likely organism. Pyrosequencing is sequencing by synthesis, so it generates 20 to 50 base sequences at a time, and that's good for short sequences. Automation allows many sequencing reactions to occur simultaneously. Then we have next generation sequencing, and this is the most common sequencing method. It offers high throughput and accuracy to sequencing via automated platforms. Variations exist depending on the specific method used, runtime, cost, and platform. Microarrays and nanoarrays. So DNA microarrays is a grouping at the micron level of DNA molecules attached to a solid support. So silicon chips, glass, plastic, often referred to as a DNA chip or gene chip. It provides the potential for investigation of gene expression from an entire organism, and it's used to analyze transcriptional levels of genes during a particular disease. So is a gene actually being transcribed by the organism? Nanoarrays, also known as nanochips, has molecules placed on the surface at defined locations with nanometer spatial resolutions. It's being developed for genomics and proteomics, such as organism ID, and they're more sensitive. Proteomics is the study of proteins on a cellular level. It can determine protein expression in certain diseases and conditions, like cancer, genetic diseases, or microbial infections. Proteins are identified in certain disease state and may become targets for new laboratory tests. Metagenomics, developed as a means to identify non-culturable microorganisms. The identification of microbial genomes from mixed populations using molecular techniques. You may find organisms underlying disease that have not yet been identified. Both sequencing and gene expression methods have been used. It has great application in environmental studies.